So, Mr. Jacob, this is what we are trying to do. Forget about all this. Let me go to new strategy. And so I want evidence from RNA-seq, and I particular about an experiment here. This trovozoite and Tison transcriptomes. So instead of going for differential expression, I chose percentile. That is the percentile of expression. So when you look at this interface, you see I'm interested in the sense, which is the forward strand, right? So I chose my sample to be trovozoite. You can change this. I want minimal expression percentile. The other one I use 70. Let me leave it at 70. A maximum expression to be 100 percentile. Matching any, leave all this by default. And I want my gene to be protein coding. So I have 1693 genes. So from this, I added this step. So I want all my genes to be a reductase. All my genes to be a reductase. I want my gene, so I use the term text, and I want reductase from Plasmodium falciparum. So I have my wildcard reductase. I run the step. So I have 84 genes from this. Also, I want my result to be a signal peptide. Then I can type my signal here. Signal peptide. Still from P falciparum. So I run this. So I eventually have 6 genes resulting from this set. So when I look at the 6 genes, the first 3 caught my attention: endoplasmic reticulum, chaperone, GRP170, exporter protein 3, and Hitchcock protein 70. So from this, I open this gene page. The gene page. The first thing I'm interested in here is my link out, which I'll be able to check for more annotation. Link out. So I check it on Uniproc, and at the same time TDR5 target database. So from my Uniport, so much interested in the Go annotation from Uniport to look at some of the functions of these proteins as predicted. Now I discovered that I have a whole lot of results from my Go annotation here. So when you look at this, the cellular component of these genes, you found this in the apicoplast, and also in the endoplasmic reticulum. So I once asked Mabel the peculiarity of apicoplast and this endoplasmic reticulum to the survival of the parasite. You can also find this one of the molecular function in ATP binding, which is very, very peculiar, important to the survival of the protein, of the parasite rather. So you can go through all these, go down as well, and look at 3D structure predicted by AlphaFold. From what we have, yes. So when you look at the subcellular location in apicoplast, you see the location of the gene you are interested in inside the cell. Can you see it?
Yeah.
So having these terms or functions from our gene ontology, especially this ER and apicoplast, at the same time, the molecular function of ATP binding make the gene very peculiar to survival. That is not the end. So when I check the TDR target database, yeah. So I have the details of the gene as well. Before my gene ontology, let me look for the expression first. Yes. So look at the upregulation of this particular gene in percentage. From 80 to 100%, you find it in all these stages. Intracytic 0 hour, 8 hours, 16 hours, 32 hours, 40 hours, 48 hours. Also find in oocyte and the ring stage. Do you get it? So if you can find this particular gene upregulated in this stage, from 0 hour to 40 or 48 hours in triatrocytic stage, it makes the particular gene indispensable. You know, if it can have up to 80 to 100% or percentile upregulation in these stages of this 48 hours. Do you get it? So it makes it indispensable. Also, it's upregulated in 24 hours of this, though in lesser percentage, 60 to 80. But once you have it in this form, you know that your gene is highly regulated in this intra-heterocytic stage, right?
Yeah.
And you know what intra-heterocytic stage is? That is the blood stage, right?
Well, yes.
Remember, we start our search from trovozoite, right?
Yes.
So you can see that. And also, I also made mention of different templates that exist in PDB. There are a whole lot here. You can see them. If you check any of this ID, it will take you to RCSB for you to do selection, probably depending on your array, your sorry for that. Okay, yes. So depending on the criteria, you have to choose using the RCSB. So as you check on UniProt, also endeavor to check on the TDR5 target database. So moving forward, let me check. You can check gene ontology. Where are you? Okay, you can check the gene ontology. This is the ID. Oh, this network is terrible. I haven't closed it. This is not good. Okay, I have it open somewhere. Oh, it's not okay yet. Crystalline structure, model 10, autologs 41. So let's go back to the UniProt. Yeah, I showed the detailed alpha-fold structure earlier, and I also mentioned the model confidence. You have more of blue, which I mentioned before the end of our internship. We'll run some R on Python script to check the health of the protein that we are going to work with eventually. So this is a simple way to build a strategy that you might want to use. So after going through the entire gene page, then you will know whether gene is essential or not. So for me, I rather these first three, iterate over the first three and see how essential those genes are. And from there, don't
I saw that heat shock. Sorry to interrupt.
No, it's all right.
I wanted to say that I saw that heat shock protein 79.
70.
I saw it in one walk that I saw online
Okay, then
when I was.
then you can let us have the article and read. So don't also forget that
So
reminds
someone did. It's actually a very important that heat shock protein.
So don't forget that you also have to check look out for the literature, right? I already mentioned that. So check literature. Here we have how many? 5 literatures. No, still loading. Health literatures. A protein interaction network of malaria parasites. Genome sequence of the so an exported protein interactive virulence determinant. So you can see this particular one. So you can see now important the literature is. So once you read, it will give you more positive confirmation that your protein is very essential. So I'm looking at this exported protein 3 now. I can check the TDR database and check the UniProt as well. You can check as many as you want. Check for genome annotation on SCBI as well.